In this video, I'm going to be talking about photosynthesis. Photosynthesis isn't only important for plants in making food, it is also important for supporting all the organisms in an ecosystem. Photosynthesis is actually a really amazing process, and many times when we think about photosynthesis, you might think like, oh, that's cool. But when you really think about what plants are doing, it's pretty astonishing. They can actually use the carbon dioxide in the air, take that in through their leaves, take water up through their roots, and from just carbon dioxide and water, they can make food. And not only do they make food, they can make all the material that they need to grow and make up their cell walls and everything. Could you imagine if all you had to do was breathe and drink water and you could grow to be the size of a redwood tree? Let's look at this equation for photosynthesis. Carbon dioxide is coming in as well as water. They're rearranging the atoms here. And what's happening is that these hydrogen atoms are attaching to the carbon atoms from the carbon dioxide, and it's creating carbohydrates like glucose. So they combine these atoms together to get glucose or other forms of sugar or carbohydrates. And in this entire process, another thing that happens is that oxygen is released. Photosynthesis occurs in organelles called chloroplasts. And if you actually look at a plant leaf under a microscope, you'll see many cells making up that leaf. So here shown in the white that I'm outlining is actually one of the cell walls of a cell. Inside the cell, you'll see many little green dots. Each one of these tiny green dots is a chloroplast. So you can see that each cell is composed of many chloroplasts stuffed inside of there. These chloroplasts are what give plants their green color. And if we were to actually look into one of these green little dots here, it would look something like this. And you can see that the chloroplast has two membranes. It has an outer membrane and an inner membrane. There's also fluid that fills the chloroplast called the stroma. And the chloroplast has these stacks within it. These stacks here are covered by a membrane called the thylakoid membrane. So the chloroplast itself consists of the fluid, the stroma, and these thylakoid membranes. Photosynthesis is going to occur in both the stroma and the thylakoid membrane. Photosynthesis actually consists of two major stages. The first stage of photosynthesis is called the light reactions. One of the reasons it's called the light reactions is that this is when solar energy is absorbed by the plants and used. The light reactions occur in the thylakoid membranes. So they occur in the stacks within the chloroplast. And a really important thing that happens during the light reactions is that the water, which plants absorb through their roots, actually gets broken down. So the plant splits the water. So remember, water is just an oxygen atom attached to two hydrogen atoms. If I split this, and let's say I broke these bonds here, what am I left with? I'm left with oxygen atoms and some hydrogen atoms. So whenever a plant splits water, the oxygen atoms are released as oxygen gas. So that's where the oxygen that plants give off comes from. But what happens to these hydrogen atoms? When water is split, these hydrogen atoms are actually carried out into the stroma by this molecule. You don't need to know the name of this molecule for lab, but the molecule is called NADPH, and it's carrying these hydrogen atoms and also using some energy. During the Calvin cycle, plants use the hydrogen atoms that came from the water when it split, and they add it to carbon dioxide. So carbon dioxide enters the chloroplast, hydrogen atoms are added to it, and what does that make? Well, that makes a carbohydrate or a type of sugar, such as glucose. Glucose is made up of 6 carbon atoms 12 hydrogen atoms, and 6 oxygen atoms. So all I did in order to make glucose was add hydrogens to a molecule that just had carbon and oxygen atoms in it. So again, just an overview, during the light reactions, water is split, oxygen is released, the hydrogen atoms from water are carried into the Calvin cycle, they're added to carbon dioxide, and that's how we get sugar. That's photosynthesis. But many people have the misconception, they think that plants are taking in carbon dioxide gas and the carbon dioxide gas is turning into oxygen that's being released. But what's happening here really is the oxygen's coming from the oxygen atoms in the oxygen gas that plants are releasing is coming from water. And the sugar is what's actually coming from the air, from carbon dioxide. So it's just something, just to think about, because it's kind of fascinating that plants are making food from air. Like, I just think that's just as crazy to me. Now let's talk about the type of light that plants use for photosynthesis. Visible light, like the colors of the rainbow, are actually part of what's called the electromagnetic spectrum. The electromagnetic spectrum is a spectrum of different types of energy, like gamma rays, X-rays, ultraviolet rays, and so forth. And these types of energy all move as waves. So they're going to move like this. Light itself is made up of a particle called a photon, and that photon travels through the air as a wave. What makes these different types of energy different is based on the amount of energy that's present. So some like gamma rays, for example, are high energy, whereas radio waves are very low energy. Visible light kind of falls in the middle, and this is what plants use for photosynthesis. One interesting thing is that the longer the wavelength is a distance between these peaks here. The longer the wavelength, the lower the energy. The shorter the wavelength, the distance between the peaks, the greater the energy. If that's hard to remember that this is an inverse relationship, just imagine that you're standing here and this wave is coming towards you. If the wave has a very long wavelength, the peaks are hitting you really slowly, so it has less energy. However, if the waves had many peaks close together, so a short wavelength, you would get pounded by those waves really frequently, so it has a lot more energy. That's just kind of how I always remember it. Longer wavelength, less energy. Shorter wavelength, more energy. Well, then what gives different colors of light their different color? Well, it all depends on their wavelength. So for example, red light has a very long wavelength, so it has relatively less energy than violet light. Violet light has a very short wavelength. And wavelength is actually measured using units called nanometers. And we can actually measure the distance between each of these peaks, and that allows us to know what color of light it is. When we're talking about white light, like the light that you see coming down from the lights in a room or from the sun looks white when the light is shining down. Well, actually, what white light is it's composed of all the different colors of light. And each of these different colors of light, again, has a different wavelength. One of the reasons you see a rainbow is because the white light is bouncing off of water particles in the air, and it's separating out the different wavelengths of light. And that's why you see rainbows after a rainstorm. In order to understand photosynthesis, you first have to understand how light behaves. And there's 3 different things that can happen to light when it hits an object. One thing that can happen to it is that light energy is absorbed. So all the light that hits the object is actually absorbed by the object. And so it's not visible to us. The light energy, the photons, get captured by that object and you don't see it. So plants can only use light that is absorbed for photosynthesis. If they can't absorb a certain wavelength of light, then they can't use it for photosynthesis. The other thing that can happen to light, if a wavelength of light comes down to this object and it bounces off. So the photon, the little particle of light, is not absorbed. If a wavelength of light hits a plant and then bounces off, it can't be used for photosynthesis because they can't harness that energy. Another thing that can happen to light is that it could actually hit an object. The photon can squeeze through and be transmitted. So anything that's transparent, like if I was holding green cellophane up and you could see through it, that means some of the light has passed through it. Now that you understand the properties of light, let's talk about why you see the different colors that you see. When you see an object that appears white, it appears white to us because all the wavelengths of light are hitting it and all the different wavelengths of light are bouncing back towards our eyes. And our brains interpret that as white. So white, again, is the reflection of all colors of light. If we see something that looks black, what is happening is that that object is actually absorbing all colors of light. So none of the wavelengths of light are bouncing back to our eyes. And that's why we interpret it as an absence of light or black. However, when we see something, for example, that looks red, what is happening here is that all the wavelengths of light are hitting that object, and all the wavelengths of light are being absorbed by that object except the red wavelength. That red wavelength is bouncing back to our eyes, being picked up by receptors in our eyes, and then our eyes are seeing that object as red. So once again, something appears red because all colors are absorbed except for red. Red is being reflected by that object. So why do plants appear green? Well, plants appear green to us because they are not very good at absorbing green wavelengths of light. So the green wavelengths of light are reflecting off the plant and being perceived by our eyes. Well, how do plants actually absorb solar energy? Plants have molecules in them called pigments. Pigments are a wide range of molecules which give color to something. Pigments can be found in plants, for example, in their thylakoid membrane. That's what gives plants their green color. However, we also have pigments, for example, in our skin, right? We have pigments in our skin that give it their brown color. We have pigments in our blood that turns our blood red. So pigments can be found in many different places, and they're just pretty much anything that gives something color. Plants have many different types of pigments. The primary pigment in plant, which is the pigment responsible for driving photosynthesis, is called chlorophyll A. It appears green because it reflects green light and absorbs all other colors of light. Again, this is why plants are green, because they have a lot of chlorophyll A in them. However, plants also have what are called accessory pigments. Accessory pigments absorb light energy. They are able to pass it on to chlorophyll A. What this allows is for photosynthesis to occur over a wide range of wavelengths. And this enables plants to absorb even more solar energy than they would if they only had chlorophyll A. Some examples of accessory pigments include chlorophyll B, which is kind of a yellowish-green color. Plants also have what are called carotenoids, which are an orange accessory pigment. Carrots have a lot of carotenoids, which is why they're called carrots. A red accessory pigment that's often found in plants is called phytoerythrin. We can actually look at how well each pigment can absorb different wavelengths of light by looking at what is called an absorption spectrum. The top of this graph is showing an action spectrum. This is a graph showing the overall photosynthetic activity or amount of oxygen produced under different wavelengths of light. And you can see the different wavelengths of light are written here at the top, and their associated color is written below that. Here we can see that most of oxygen production in plants occurs around violet light, and around maybe like the reddish wavelengths of light. But the least amount of oxygen is produced under green wavelengths of light. The bottom graph here is showing an absorption spectrum. You should be able to interpret absorption spectrums for different pigments. What this graph is showing you is what colors or wavelengths of light are absorbed best by each pigment in a plant. Each line here is representing a different pigment. So let's talk about how we can interpret an absorption spectrum. First thing, remember each line represents a different type of pigment. The x-axis here is showing us the different colors of light or their different wavelengths. The y-axis is showing us the amount of absorbance happening in that specific wavelength. The higher the peak, that means a higher absorbance, which means higher rate of photosynthesis can occur. The more of a certain color of light that's being absorbed, the more photosynthesis that can happen under that wavelength. Lower peaks, or where there's no peaks at all, for example, like right here, if we're looking at chlorophyll A, which is shown by this dark line, right here there's no absorbance happening. What that means is that since that light's not being absorbed, for example, blue light is not being absorbed by chlorophyll A, and green light barely is. Those colors of light are therefore being reflected. So if it's not being absorbed, if it's low here, it's being reflected. And this is why chlorophyll A looks kind of like a greenish color or blue-green color, because look, it can't absorb blue-green light at all. So again, chlorophyll A, this is its absorption spectrum that you can see here. It's a primary pigment in plants. It absorbs violet light the best, because that's where its peak is. And it reflects green light, giving it its green color. Accessory pigments, as I stated before, allow photosynthesis to happen over a wider range of wavelengths. If chlorophyll A was the only pigment in a plant, then no photosynthesis would be happening over this range here, because notice how this very little light is being absorbed. However, having all these accessory pigments available here, you can see there's many peaks within that range. So they're broadening the amount of absorbance that's happening. Examples of these accessory pigments include chlorophyll B, which looks like this. What light can chlorophyll B absorb the best? Well, that's around the violet blue range right here, around 450 nanometers. And it's not very good at absorbing greenish yellow light, which is why chlorophyll B looks like a greenish yellow color. Carotenoids cannot absorb yellow, orange, or red light, which is why they appear orange. However, they can absorb some bluish green light, which allows photosynthesis to happen over that range a little bit. Physoerythrin, this is a red accessory pigment. Notice it can't absorb any red light down here, and this is why it appears red. But physoerythrin is really good at absorbing green light. So a plant that has a lot of this pigment in it can increase the amount of photosynthesis happening under green light. Let's summarize everything that we just learned. Plants have molecules in them called pigments that absorb light. The primary pigment in plants is called chlorophyll A. However, plants also have accessory pigments like chlorophyll B and carotenoids that help broaden the spectrum of light that plants can use for photosynthesis. Plants are not very good at absorbing green wavelengths of light. Because of this, plants appear green to us. And because of this, plants have a lower rate of photosynthesis under green light than they do in wavelengths of light like violet light or red light. Let's do a sample problem to make sure that you understand how to interpret these absorption spectrums. If a pigment had this absorption spectrum, what color would this pigment be? Hopefully you answered violet, because violet light was not absorbed by this pigment, which means it was reflected. Well, how do we know that plants have all these different pigments? Well, you can actually observe these different pigments in plants by doing what's called a thin layer chromatography. A thin layer chromatography is a method of separating out plant pigments based on their solubility, or in other words, how well they dissolve in a specific solvent. And the way you will do this is we will actually crush up spinach leaves. And when we crush up the spinach leaves, we'll get this green extract from that. You will add that green extract to a piece of paper, like make a dot of it, and then you'll put it into a test tube. At the bottom of the test tube, there will be a solvent in there, kind of like acetone, and this solvent is nonpolar, and it will move up the paper strip and be absorbed up the paper strip, and it will carry the pigments up the strip. The more soluble the pigment is, or in other words, the more nonpolar it is, the further up it will actually be carried up the strip. So let's look at this. Here's the extract. You can see the solvent moving up the strip. Because the solvent's nonpolar, anything else that's nonpolar will be carried by it easier. So the more nonpolar the pigment is, the easier it will be for that pigment to travel up the strip and be carried by the solvent. And what you can see here is all these pigments actually separated out so that you can see the yellow and the darker greens and the lighter greens of those different pigments in a plant. You might get results that look something like this. And this is the solvent front is the distance the solvent traveled up the strip. And then you would want to mark where all the different pigments were that moved up the strip. The start line is where you first applied the extract. One of the benefits of doing a thin layer chromatography is that you can look at the distance and the color the different pigments moved, and that can help you actually identify what pigment it is within a plant. Another thing that you will have to do when you look at a thin layer chromatography strip is calculate the ratio of how far the pigment traveled compared to the distance the solvent traveled up the strip. And we call this an RF factor. So again, the RF factor is simply calculated by measuring the distance the pigment traveled from the start divided by the distance the solvent traveled from the start. And you will have to calculate that for each pigment that you see move up your strip. One thing to remember is that each pigment has a different RF value between 0 and 1. So if your RF value is less than 0 or greater than 1, you did something wrong. The most soluble pigment travels the farthest up the strip, so it's going to have a higher number, or a number closer to 1. If we were to calculate the RF for the yellow pigment here, since it moved 5.5 centimeters, we would take 5.5 divided by 6, which is the distance the solvent moved, and we would get 0.917. The orange pigment, we would do 3.5 divided by 6, get 0.58. Light green, 3 divided by 6, get 0.5. And the dark green is 2 centimeters divided by 6 is 0.333. The yellow pigment was the most soluble, and the dark green pigment was the least soluble. In the second part of your experiment that you will be conducting is you'll be measuring photosynthesis. And you'll be measuring photosynthesis under white light and green light. We know that plants don't absorb a lot of green light, so we would expect that photosynthesis would be less under green light. In order to measure photosynthesis, you're actually going to be measuring the amount of oxygen produced. And the way you will do that is we will use aquatic plants called Elodea, and we will put them in a test tube. The gray solution in this image is actually sodium bicarbonate. Sodium bicarbonate is a great solution to use because it absorbs carbon dioxide gas, and it will take that gas out of the air so we don't have to worry about carbon dioxide gas affecting our measurements here. We want to strictly be measuring changes in oxygen. So how will we measure the total amount of photosynthesis? What we're going to first do is measure the amount of the net photosynthesis happening. And we will place our Elodea into a test tube. We'll place a cork on that with a pipette in it. Some of the sodium bicarbonate solution should move up the pipette to start off the experiment. and you will have a little bit of air in the test tube. Next, what you will do is you will mark the initial level on your pipette. And what you will do is you will place a beaker of water between a light source and your Eladia plant. And the reason we put this beaker of water here is to help absorb some of the heat energy produced by the lamp, because we don't want that affecting the pressure, the air pressure inside our tube. After you turn the light on, the plant should start photosynthesizing more because it has more light energy. And what you will observe is that the plant, remember, the plant's taking in carbon dioxide, but it's taking carbon dioxide in from the solution. All that carbon dioxide is staying in that solution. And it's taking carbon dioxide, but it's also releasing oxygen gas. The oxygen gas is going to accumulate in this airspace here, because the oxygen gas does not stay in the solution. And as the oxygen gas starts to accumulate more and more, it's going to push this sodium bicarbonate solution down here, and it's going to cause the pressure in here to rise, so the sodium bicarbonate solution should move up the pipette. And after 10 minutes, you will measure how much oxygen was produced by measuring how far up the liquid traveled. And let's just say our measurement was 32 millimeters. So we'd write down 32 millimeters per 10 minutes. However, the problem with this measurement here is that we're not taking into account the oxygen that plants are taking in when they use cellular respiration. So remember, plants are not only photosynthesizing, they are also using cellular respiration. They use photosynthesis to make glucose, but as we know, glucose cannot be used directly by cells for energy. Instead, cells always have to convert glucose to ATP. So plants are also using this cellular respiration, and in cellular respiration, plants are taking in oxygen for that process. So what that means is that this measurement of photosynthesis is actually lower than what it should be, because the plant actually was taking in some oxygen for cellular respiration. So we need to figure out how much plants are taking in for cellular respiration so that we can add that back to this number up here. In order to measure cellular respiration, what you're going to do is cover the tube up with aluminum foil so no light can come in. Now the plants can't photosynthesize, they can only use cellular respiration. And then you're going to wait 10 minutes, and what should happen to the volume in this tube? Well, it should go down because they are using oxygen for cellular respiration. Let's just imagine that it was 1.5 millimeters that they used. Then we would just add these two numbers together, and that would give us the gross photosynthesis, the total amount of oxygen that the plant produces in 10 minutes. We can then calculate the rate of photosynthesis in millimeters per hour by multiplying this number just by 6, and that would give us 201 millimeters of oxygen per hour that a plant, this tiny little plant, would be producing. To measure photosynthesis in green light, you're going to use the exact same procedures as white light, except that you're going to use green food coloring and add that to this beaker so only green light is going through, and you're going to wrap the tube in green cellophane so that only green light can enter that tube as well. And then this will allow us to measure the effects of only green light being absorbed by a plant. All right, that is the end of the overview of photosynthesis.